Good morning. We're going to get started. My name's Catherine Nevins, and I'm here as a pro tem for Dean Pamela Irwin, who is unavoidably delayed and gave me a quick call, and I was thrilled to be able to step in for her. Welcome. This is one of our primetime presentations that the library, Bethel University Library, designates to celebrate the experiences and accomplishments of our faculty and students and staff. It is a collaborative process and experience between the Friends of the BU Library, which you can join even as a student. You can become part of the Friends of the BU Library with faculty development and also with other offices on campus. Most of the presentations are recorded and can be found in the BU Digital Library, which is just a wonderful resource for us, I think. So be sure to check the Library News and Events webpage for future presentations and the BU calendar for things that are going to be coming up. And the next actual presentation for the prime time will be on March 29th with Dr. Ripley Smith, who is a professor of communication studies. And he will be sharing a comparative study of virtues from 14 countries, which explores the I'm going to have to read this, which explores the connections between faith, place, and culture in order to assess the utility of virtues as horizons for intercultural understanding. There you go. The operative word for me is virtues, cross-culturally. That sounds fascinating. Well, today, I think most of you are here because of the opportunity to hear from Brian Hyatt and Nate Riedeman, who will be presenting on their Edgren Scholars project that they did last summer. Just a word about John Alexis Edgren. It is a name that those of us who have been at Bethel for a long time feel in our bones. He is the founder of Bethel. Long ago, he was the Bethel Seminary founder, and from that one man's vision comes the institution that you are now a part of. What is unique and special to us at Bethel is that Edgren believed, and I'm going to quote this, that the relation between teacher and students should not be that of superior and subordinate, but one of real friendship and helpfulness. There we go. In this spirit, the Edgren Scholars Program was developed in great part to encourage and facilitate student and faculty collaboration in research. So that is the program that Brian and Nate were part of. It is a process by which they receive funds and support for their research. And the project must be one that has the potential to make a significant contribution to a given field of study, and the project must reflect meaningful collaboration on research between students and faculty. This is not a TA or a research assistant. You are co-collaborators in that research. And to that end, we are here to hear from Brian and Nate, who is a biology major. Can't understand why you would want to do this project if you were in communication studies, unless South African frogs make noise. So, sorry, enough of my humor. Let's hear from them on the Xer81 gene's molecular effects on neural tube development in frog embryos.
All right. Thank you for everyone that's shown up. So Nate and I have broken this up. I'll start at the beginning of the talk and then turn it over to Nate. And as Kathy said, this has been a collaborative project. I'm going to do some introduction, tell you about how we got started and things, and Nate's going to take over, and then he's going to tell you how he has integrated a lot of his thoughts and a lot of his experiments into this. And the project is really sort of a new direction of research that I've done and students with me have done. So Nate's sort of the first person to ask this specific question, which you don't know yet because I haven't told you, but this specific question that we were looking at. So we built a pretty nice base. We think that students in the future will be able to participate in and continue to move the project. So role of Xer81 in neural tube formation. So Xer81 is a gene, and I'll talk about that in a minute. I didn't know that they were going to use this picture to advertise, but I put it up here to show the star of the show here, the frog. Xenopus laevis is an African clawed-toed frog. It's just a model organism to study developmental events. This is an adult female there. We use females to get the eggs, males to get the sperm to create the embryos, and that's what we really look at, okay, the very early developmental events. Give you an idea sort of what they look like here. So this is a bunch of embryos. They're 16-cell stage, meaning they only have 16 cells at this stage. You can sort of count them: 1, 2, 3, 4, 5, 6, 7, 8. And on the other side, there's another 8 of them. They are about 1.5 millimeters to 2 millimeters in diameter. So I brought my ruler here. I can't even, it's really, really tiny. But with microscopes, we can see them and look at them. So not very big at this stage when we're starting to do our experiments. So Xer81, what is it? It's a gene. It's a gene that codes for or has information that is used to turn it into a protein that's called a transcription factor. I'll talk about that in a second. Genes have funny names and they have history to their names. So I just want to say, what does all this mean? So X stands for Xenopus, ER stands for ETS for the E, related for the R, and number 81. I'm not sure where anyone came up with 81. So then it's, well, what does ETS mean? So ETS means E26, transformation for the T, specific for the S. Still doesn't explain what it is. Well, E26 is a leukemia virus. So it's a gene that was first discovered in the leukemia virus. All right, and it was involved in what viruses sometimes do, they transform cells. So it's E26, this virus, transformation specific. Still doesn't tell you what it does, it just tells you where the name came from, okay? So it's this gene, it's a transcription factor. What do transcription factors do? Okay, so to do this, we have to sort of shrink down and think about the DNA on the inside of our cells. So humans have 3 billion letters, or 3 billion pieces of information, okay? Which we just designate by letters here to designate the type of chemical that those letters represent. And so there's 3 billions of these. Frogs approximately about the same amount. A lot of organisms have a little more, a little less as far as numbers. And those letters are put into a particular order called genes. And that information contained in the order of those letters is used ultimately to make a protein, what I call a mini machine. So we have these little machines in our cells that do jobs. Okay, they carry out functions. And so Xer81 is coded for by a bunch of letters. Ultimately, it gets made into a protein. The type of protein it is a transcription factor. Right? And the job of transcription factors then is to regulate or control the event of taking the information and turning it into a machine. Okay, so Xer81's job is it regulates certain genes as to whether or not they make their product. Okay, so it's a regulator. It controls. Now the question is, what genes is it controlling? Okay, and that's what we don't know. All right, so that's part of what is involved in this project, is to try and discover what genes is Xer81 actually controlling. It may be a few, it may be many, and there may be other factors helping it do its job. So, how did we get into this project? I said that this is a little bit different than what we've done before. I'm really interested in how lungs develop. It's what I've been studying for nearly a couple decades now, I guess. And we first discovered Xer81 as a gene that's expressed in Xenopus or in frog lungs. And so frog lungs, So to show you, so this is about a 2.5 to 3 day old embryo. So I showed you a 16 cell embryo. That's about a 3 hour old embryo. This is about a 3 day old embryo. And you can see the eye there in black. This is the digestive system here. The mouth will be forming here. And the lungs sort of sit right on top of the digestive system. If we color them, you can see them here in this bluish color here. This is looking right at the back of the embryo. Here's the eyes here. You're looking right down on the back. And the lungs actually form sort of this crescent or this U-shaped structure. And so it turns out then that Xer81 is expressed in developing lungs. And this is what got us interested in this. And so this is just a picture here showing the lungs coming off of the esophagus and going into what develops as the stomach and the intestinal tract there. And so this paper is the first paper I saw where Xer81 was expressed in the lungs. So it's listed here without the X, just as ER81. So here's the digestive tract, and you see this blue here. It's like an upside down Y. This gene is expressed, or it's being made into its product in the lung tissue specifically. So we got interested in that, and several students for a few years have been trying to discover what it exactly is doing in the lungs. And it turns out while we were doing the studies last year, before Nate started, that we discovered that if we suppressed the ability of Xer81 to be made, so we tried to eliminate it from the embryo to see what happens, and we were interested in what happens in the lungs, what we ended up seeing is a unique neural tube phenotype. Okay, so we saw something unexpected, but something really interesting. So what is the neural tube and why is it important? So the neural tube is the precursor to our brain and spinal cord. Okay, so it's a precursor to our central nervous system. And it is actually a tube. It rolls up, forms a tube. Ultimately, we get our vertebral column surrounding it, protecting it, our skull surrounding and protecting our brain. But it starts embryologically as what we call a neural tube. And it forms in, say, a unique way, but developmentally it forms in a very interesting way. So it starts as a flat sheet of cells on our back. So if you just sort of think of our adult system as a back, the embryo has a back. You have this flat sheet of cells. And what it does is it folds up on the sides and it rolls all the way up, and then it fuses or sticks together until you get cells on the outside and you get a lumen or an opening on the inside. Okay? So it's a very dynamic process. Let me show you a picture of this is electron micrographs, so highly magnified images of chick neural tube development. So here it is, sort of folded up, hasn't closed in the middle yet. Here's another picture of it folded up, hasn't closed in the middle. And here's a picture of it all the way around, so it's fused, or curled up and fused all the way around. And these things called somites then will help form the vertebral column, or the bones that will help surround it and protect it. And so what happened? So we did some experiments where we suppressed the ability of the embryo to make Xer81. So now it's missing. So we're asking, what happens if the regulator is not there to regulate? All right? And what ended up happening is these are the controls here. And so the heads are down here. And you can see this sort of sharp line. And what's happening there is the neural tube has folded up and it's just starting to fuse. And you can sort of see the center of it there. But when we did this, it's called MO, and we'll explain, Nate and I will explain what that means. When we did this, the folds are now far apart. You can see there's some variability in the phenotype or the outcome. In this embryo, you have a big open neural plate, and the folds are really far apart. They started folding up, but they didn't close. And they eventually do, for the most part. But they're very delayed. And you can see in this one, they're a little closer together, and this one, they're even closer together. Okay? But we see this phenotype, and so the neural tube is not closing. And we also put more of Xer81 in the embryos, and it turns out if we put more in, they close, and we're still trying to determine if they sort of close with extra ridge or not. It's a little harder to tell just looking at it, but we're also exploring what happens if we put more of that gene in. And here's just another picture of the embryos a little bit later. So normal embryos, here's the head region at top, and you can see they start to close here, but in the most anterior region where your head would be, it's still open. in those regions. So the neural tube is really delayed in closing properly. All right, so why is this important to look at? So the neural tube not closing falls into a class of neural tube defects. Okay, it's a defect in the development of the neural tube. You can imagine that would have problems. It's estimated to occur in humans, at least, in about 1 in every 1,000 pregnancies, and there's different reasons for it. And it usually is classified, and classified in a number of different ways, but 3 sort of ways classifying it from the most anterior of the head down to the most posterior, the bottom of the spinal cord, as anencephaly, which is just the absence of brain formation. Okay, so the brain doesn't form because the neural tube didn't develop properly there. I have trouble saying this word. Rakish, which is open, sort of in the middle of the back. You don't get the vertebral column closing. The spinal cord and the nerves are exposed. And spina bifida, which would occur a little bit, as most people traditionally think, further posterior in opening, and the spinal nerves are exposed. So it's a big problem if this does occur, if neural tube doesn't develop properly. And so just a little image here, just sort of showing where these things happen. So anencephaly is absence of brain happening, rachitis in about the middle of the back, and spina bifida further posteriorly. So we knew a couple of things going into the study last summer. We have this gene that when we suppress its expression seems to cause these neural tube defects. All right? So then the question is, why? And so it was a really open question. We could have approached this in a number of different ways. And we chose to approach it with the fact that we know that there's a certain class of molecules that are necessary or are involved in the process of closing the neural tube. Okay? So cell adhesion molecules, okay, are molecules, they're proteins that do exactly what their name says. They help cells adhere or stick to one another. Okay? And so it's known that several different cell adhesion molecules are necessary for proper neural tube closure. So we know cell adhesion is necessary for neural tube closure. We know we have this gene when we suppress it, has disruption in neural tube closure. So the question we asked, the hypothesis we came to, was will inhibition of Xer expression affect the expression of cell adhesion genes? In other words, we know these two things affect neural tube defects. Are they causative? Does one influence the other? All right? And we don't know. Right? We went in having no idea, but that was the hypothesis we went with. Okay, so what are cell adhesion molecules, then, just very quickly, and why they're important? They're just molecules, and you can see they're joined here, that stick together and help cells stick together. The next image is a little bit better here because it shows the cells. So you have a cell here in gray and a cell here in gray. You can see in pink here, they express, or they have these proteins that are on the outside of the cell and are stuck inside the cell as well, and they interact or they stick or adhere to one another. That then helps the cells stick or adhere to one another. Then you have other classes of adhesion molecules that help the cells stick to what we call extracellular matrix, or stuff that's outside of the cell. And these then are important in this process. We know that cell adhesion molecules are important for cells to stick together because if you put here, we'll look at this one, N-cadherin, or in the middle one, E-cadherin and E-cadherin. You put E-cadherin in the red cells and E-cadherin in the green cells, these are cell adhesion molecules, and you mix the cells together, the cells all stick together, green and red stick together equally. But if you put two different types of cell adhesion molecules, E-cadherin in the red cells and cadherin 6b in the green cells, the red cells stick together preferentially and the green cells stick together preferentially. So our cells use specific cell adhesion molecules to stick specifically to some cells and conversely then to not stick to other cells. So sticking together is important, but not sticking to other things is just as important. Oh, got an extra one in there. Okay, so how does this relate then to neural tube closures and cell adhesion? Well, it's not drawn in here, but this looks like just one big drawing, but it's made up of hundreds of thousands of cells in here. All the cells in the neural tube in the light blue then need to stick together because as they're curling up, as the top cell curls up, it needs to stick to the next cell, to the next cell, to the next cell to exert a force to pull the whole sheet up. All right? The cells in the bottom half here need to stop sticking to the stuff underneath them. Otherwise, if they're stuck there and you try and pull, you can't. So I sort of think of if you have some linoleum or maybe a big piece of carpet stuck on the floor, right? And you want to curl that up. Well, you got to take care of the glue underneath the carpet in order to curl that up. Okay, so that's the adhesion, taking away the adhesion of the cells to the bottom, all right? But it's really not like a carpet, since it's made up of a number of cells. It's more like tiles put into the floor, all right? So now if you think of taking the edge of tiles and folding them up, you're going to crack all those tiles, right? And all the grout between the tiles, they're not going to stick together. So the cells not only have to lose the adhesion from underneath the fold, but then they have to create adhesion. The tiles have to adhere to one another next to it, so the tile on the outside can pull the tile next to it, and next to it, and next to it, and so that they can create this fold, okay? So that's what it is. That then was our hypothesis, right? Okay, cell adhesion is important. This Xer81, when we suppress it, has this effect. Let's see if we can see if those two events are related. Okay, so how do we do this? Just very quickly, and then I'll turn this over to Nate. So we suppress XER expression via what's called antisense RNA. And what does that mean? So going back to this piece of information up here, okay, so DNA is a piece of information and actually is turned into RNA, which is another piece of information. And those letters then cause the creation of this green box, which is the protein, okay? And so in this case, the green box would be the Xer81 protein, which is a transcription factor that controls something. So we want to stop that process, okay? We want to see what happens if there is no Xer81. So what we do is we use what's called antisense RNA. Just quickly, antisense RNA binds to the information piece, or the RNA, and it inhibits this event from happening. Basically, sterically, it gets in the way of the process. Okay? It doesn't allow the process of making the protein occur, so it stops it. All right? And so we used an antisense molecule called the Morpholino. I'm just going to sort of gloss over this. The reason we use this is it's a chemically synthesized molecule, and it has a long half-life. It doesn't get broken down by the cells. So it allowed us to put this into the embryos, and it would last a long time rather than a short period of time. And how we did it? Microinjection. So a physical injection. I'm going to skip this slide. Physical injection into the cells. So this is the same picture of those 16-cell embryos. Just a couple of arrows here. You actually poke a needle into that cell, inject a small amount of that antisense molecule, stick into another cell, inject a small amount. And we can do this anywhere, well, at any point, but we've done it from 4 cells all the way up to 16 cells. So we can do it early, affect more of the embryo, do it a little bit later, and focus it on a smaller part of the embryo. Yeah, and I think that's it. So then when we do that, we select the embryos that have this neural tube phenotype, and then Nate's going to tell you what we do with them afterwards.
All right, so just to start, I want to do a quick recap of some of the things that Dr. Hyatt said. These are probably our most basic and most essential information that you need to know for this process. Neural tube closure is essential for the development of the central nervous system. That's what makes this whole research project important. We can relate any findings to this to similar genes in humans, and so we can learn more about the development of the central nervous system. Neural cell adhesion molecules play a role in this process, so that is what we are studying. We want to examine the effects of the XER gene on the neural cell adhesion molecules. We do know that when XER is knocked down, the neural tube does not close in the embryos until very later stages, so it's very delayed. And even with it, it doesn't always completely close. We actually did have death of a lot of the embryos that didn't make it to later stages, possibly due to the fact that the neural tube did not close properly. So our hypothesis was essentially we wanted to see how XER was related to the neural cell adhesion molecules. Since the neural tube did not close, we originally had hypothesized that there would be less expression of neural cell adhesion molecules when Xer81 is knocked down. So since this is a brand new project here at Bethel University, we needed to make tools that we did not previously have. To make tools, we needed to start by cloning genes. And the genes that we needed to clone were the neural cell adhesion genes. And so the list of genes is listed up there. We listed N-cadherin, F-cadherin, focal adhesion kinase, nectin-2, and TAF1 beta. These are all just genes that we had found from previous literature that are involved with the closure of the neural tube. They're all expressed in the Xenopus embryos. Another gene that we had looked at was the noggin gene. This is a gene that has previously been shown to cause an enlarged head in an embryo when overexpressed. We did let some of the Xenopus embryos go to a later term, and those embryos did have a larger head with a wider gap between their eyes. So we figured that noggin would be a good gene to also chase. But we also did find that it is expressed in the same time and the same place as the Xer81 gene, so that we think that there may be a correlation there as well. So we made these tools. We used a technique called PCR, polymerase chain reaction, and we cloned the genes, and we inserted them into a vector. And that is just so we can stabilize the gene, and then we can use it for further experiments as well. They can actually be used for microinjections if we wanted to overexpress any of these genes. Since it is in a vector, we can microinject these genes and express it in future projects. We verified this, that our clones were successful. We ran an agarose gel to verify that all of the genes were the correct size. And then we sent them into ACGT Incorporated, and they sent back sequence results, and from there we used a computer program to put together the sequence to verify that we had the correct gene. So next I want to talk about the process of microinjections. To do this process, we had to first get eggs from the females, and so we would inject the female frogs with hormones, and this would stimulate egg growth. And then we would massage the eggs out, and then sacrifice the male and mince the testes, and then from there we would fertilize the eggs. We would allow those to develop until the 4-cell stage. Most of our injections were done at the 4-cell stage, and then some were done at the 8-cell stage. And then Dr. Hyatt performed all of the microinjections of either the PXEX vector, which overexpressed Xer81 in the embryos, or he would inject the antisense Morpholino oligonucleotides, which would underexpress Xer81 in the genes by competitive inhibition, as he had described before. We would inject on the dorsal side, so on the back side of the embryo. To do that, we would use a technique called tilting and staining. We would induce cortical rotation, which determines which side of the embryo becomes the dorsal side and which side becomes the ventral side. And then we would stain using Nile blue the dorsal side of the embryo, so Dr. Hyatt knew where to inject the embryos. Yes. So following the injections, we allowed the embryos to develop to stage 17, which is the late neurulation stage, which is right where the embryos will be closing their neural tube. And this is the same picture that Dr. Hyatt had showed before. The arrows are actually pointing to the anterior portion, so the head region of the embryo. And you can see that in the antisense Morpholino oligonucleotide case, the underexpressed embryos have a more open anterior neural tube, and also more open than the controls in the posterior region as well. So once we collected these embryos at stage 17, we would extract the RNA, and we would use the RNA as a template to we would use the extracted RNA to make cDNA, complementary DNA, and then this could be used as a template for qPCR, which I'll explain that a little more later. And the cloned genes that we had used before, so like I said, we cloned all of our neural cell adhesion molecules, or neural cell adhesion genes. These can be used as standards for qPCR, and this helps us determine the amount of gene expression that is occurring for each gene of interest. So the way qPCR works is it allows us to quantify levels of gene expression in the embryos. So this requires a master mix with fluorescently tagged molecules and cDNA template. And the cDNA template that we use is the embryos that we had previously harvested from the last slide. Our DNA standards are standard concentrations of the neural cell adhesion molecules, and we did that in tenfold dilutions, which allowed us to quantify the level of gene expression. The way that the qPCR machine measures gene expression is it measures how much fluorescence is added when the new strands of DNA are synthesized. qPCR, when we do this, we don't get a usual data set. We are unable to use a standard test like a t-test because we get non-parametric data, which means that our data does not follow a bell curve. Therefore, we have to compare our data based off of the median. So we analyzed our results using a Mann-Whitney U-test, and this was a great test because it is perfect for non-parametric data, and it compares everything based off of the median. So now the fun part, we're going to go over some of the results that we had. We ran qPCR for all of the genes, for all 5 of the neural cell adhesion genes and for the gene noggin, and collected data. But first, we wanted to make sure that the PXEX vector was actually adding more Xer81 gene products to the embryo. So in the first pie chart here, we did not find a statistically significant difference in the amount of Xer added to the embryo, but based off the pie chart, you can see that 2/3 of the data samples that were, 2/3 of the samples that were above the median were the overexpression case. So we do see more, but based off of the Mann-Whitney U-test, it's not statistically significant. Following that, we have the gene TAF1 beta, and if you'll remember, this is one of our neural cell adhesion molecules. We found that when Xer81 is knocked down, there is less expression of TAF1 beta in the embryo. And we used a large sample size for this, and the Mann-Whitney U-test showed that there was statistical significance. So we can verify that when there's less Xer81 in the embryo, there's also less TAF1 beta neural cell adhesion molecule. Interestingly enough, when we overexpress Xer81 in the embryo, we also find less expression of the TAF1 beta. And I will talk about more theories behind that later, because it's kind of an interesting result. Then we looked at one of the genes, Nectin-2, and we found statistical significance with this one as well, where when Xer81 is overexpressed, there is less expression of the Nectin gene. And then we have the gene noggin, and in both cases, when we overexpress and underexpress Xer81, there is less expression of the gene noggin, which is also a similarly interesting case as the TAF1 beta. And then finally, we found that when Xer81 is overexpressed in the embryos, N-cadherin is also underexpressed. So, and you can see that that one was very statistically significant. We had 100% of the uninjected embryos that fell above the median, and then all of the samples below the median were the PXEX overexpression case. And then we had a bunch of conditions without a statistical significant difference. This could be due to a low sample size. These were genes that took longer to clone. They are all a larger length, which makes them harder to clone. So they weren't cloned until late in the summer, later in the research project. So we did not have a huge sample size with these, but we did not see a statistically significant difference when comparing the uninjected to the underexpressed for Nectin-2, focal adhesion kinase, N-cadherin, and F-cadherin. And we also did not see a statistical significant difference in the uninjected versus the overexpressed for focal adhesion kinase and F-cadherin. So some of the conclusions that I made based off of this data and these results and statistical analysis was that clearly, phenotypically, Xer81 does play a role in neural tube closure. If it doesn't, we wouldn't see, if it didn't play a role, we wouldn't see such a clear phenotype. Quantitatively, when we knock down Xer81, we see less expression of the TAF1 beta and the noggin gene. This is kind of contrary to what we had originally thought for the noggin gene. We thought that knockdown of Xer81 would lead to overexpression because we had a larger head. But we found actually the exact opposite. And we had exactly what we expected for the TAF1 beta, that when there is less expression of Xer81 gene, we get less expression of the TAF1 beta. So we're getting less neural cell adhesion molecules of the TAF1 beta protein being expressed. One thing that was very interesting that I noticed was that many of the overexpression cases where we overexpress Xer81 in the embryo, we found that there was less expression of all of the genes that we had tested against. So I have two ideas why this could be happening. The first is that there could be a potential threshold concentration of Xer81 that is optimal for the activation of other genes. That if we are too high above this threshold concentration, that we are lowering the expression of the genes. My other theory is that the embryos become unhealthy. We did notice that when the embryos went to a later stage with the PXEX vector added, that not all of them made it. There was less and less that made it as you got to later and later stages because the embryos did get unhealthy. Another idea with this is that, as Dr. Hyatt said before, is that there was too much sticking of the neural tube to other tissues. So if we have overexpression of XER in tissues where it's not supposed to be overexpressed, then we could have too much sticking and the neural tube fails to be able to lift from its basement membrane or from other cell types and be able to close into a functional neural tube. One of the most exciting parts of this project is that this is a brand new project and there are a lot of places where we can go with this. Our results are great, but the future work that can happen with this is unbelievable. We can go very, very many different directions. Currently, I'm working on a DNA microarray, which some of you may know as a DNA microchip. And the DNA microarray allows us to analyze all of the genes that are being expressed in the embryo. This is great because we can take an embryo that is underexpressing Xer81, and we can take an uninjected control embryo, and we can compare all of the genes being expressed in there. This would be great because in future projects, we would already know which candidate genes to go after in using qPCR, because we would know which genes are being overexpressed and underexpressed when Xer81 is being knocked down. More future work that needs to happen. I have 3 other genes that I did clone this summer. I have the gene RAS, DBA, XAGR2, and NLO. Data has been collected on these, but they were kind of funny data sets, and the statistical analysis was kind of tricky, so they did not quite make this presentation, but they will be done by symposium time, so that I will be presenting on those 3 genes as well. And then another way that this could go is that Dr. Hyatt said that we were originally interested in the lung development. And I think this would be another way to go with this project as well. Since we're already doing all of the microinjections, all we have to do is allow the embryos to develop to a later stage so that they are at a lung development stage. So Dr. Hyatt can perform the injections. We can analyze some for the neural stage, let some other ones grow later, and we can analyze those for the lung stage. And this is very important because Xer81 has been shown to respond to FGF signaling. And this FGF signaling pathway is a major pathway involved in lung development. So we can do the same experiment, letting the embryos grow to a later stage, and we can test Xer81's effects on Wnt2b, surfactant protein C, surfactant protein B, NKX, and SOX2, which are all important genes that are involved with lung development. So we can test those specific markers to see if XER plays a role in lung development as well. Since this is such a novel project, I'm very excited to see where some of the future research leads with this. And that about does it for this. I would like to say kind of a special thank you to Dr. Hyatt for working with me on this project. I learned a ton. It was very collaborative. It was great bouncing ideas off of each other, and it was very cool to see how like a new project could begin and where it can go from such a simple idea in the beginning to many complex ideas where a microarray is being run. We did quantitative PCR, gene sequencing, and cloning. We just did everything for this one project. It was great. I would like to thank Bethel University Department of Biological Services for providing us with the tools that we needed. I want to thank the Edgren Scholarship community for providing us with the funds. And then also, at the end of the summer, I did get another grant from the Tri Beta National Honor Society, which provided us with more funds so that we could do a microarray. Does anybody have any questions?
Just a quick one, Dave.
Yeah.
In all this excitement, and there's so much to do, are you going to carry these projects to grad school, or is Dr. Hyatt going to carry them on in sabbatical next year?
I think what the thing is that all of the clones were made here, and all of the clones are here. So all of the tools that we had made, this was a full summer project. I was working 40 hours a week this summer, just cloning genes. And then kind of the last month of the summer, I finally got to start doing quantitative PCR and get some data. So I'm assuming since all of the tools are here, this will be something that Dr. Hyatt will be able to continue here. And the microarray will be great because then he'll know which genes to clone next and stuff like that. Yeah.
Okay.
Thank you.